Hi everyone, it's Sophie speaking. We will start shortly and still wait for the last participant to connect. Hi everyone, so Sophie speaking again. I think that all the participants are now registered, so we will start the webinar. Please don't hesitate to use the chat box or question box if there is any problem. So we will begin this webinar around the best practices for environmental detection and monitoring of airborne viruses with Coriolis air sampler. We will start with the agenda of the session. So this afternoon in France, I'm with Dr. Florian Cohen, who is field application scientist at Bertin Technology, and myself, Sophie Dubacq, as product manager at Bertin Technology. We have three topics in this webinar, in this session. First thing, we will introduce the Coriolis air samplers. Then Florian will introduce you the tips and tricks to collect viruses in the environment of SARS-CoV-2 pandemics. And the third session will be around case studies where the Coriolis was used to collect airborne viruses. And then we will finish with a Q&A session. First thing, some rules about the webinar. So your microphone is automatically muted to avoid and minimize the interferences and background noises. So if you have any questions or remarks during the presentation, please write it in the question box. After the presentation, Florian and I will answer your questions. To start, we will present you the Coriolis air sampler. Just to begin, I would like to introduce really shortly Bertin Technologies and what his know-how in life sciences. So Bertin, it's more than 15 years of expertise in designing and manufacturing high-quality lab equipment. So we have the topic of today, which is the Coriolis Micro, an air sampler for biocontamination quality control. But not only, we also design and produce the Coriolis, which is a universal tissue homogenizer to homogenize all your tissue and analyze DNA, RNA, or protein. We also have a huge range of bioreagents, mainly with ELISA kits, and we have the INFLUENZA, which is a smart cell imaging system. In terms of cyclonic technology for airborne contamination control, we have more than 10 years of activities here. This project start with two requests we had from customers, one for the Mona Lisa project, which was an European project for pollen and allergen detection. And the second one was a project we developed with the French Army to collect and detect airborne pathogen in the air, such as anthrax. We have new two instruments available in this range of product, the Coriolis Micro and the Coriolis Compact, that we just launched this year. And I will introduce you the technical features, but Florian, after that, will present you some results we already have with this instrument. Just for you to know, the Coriolis, as you can see, it's more than 10 years of experiments, and it was validated and used all around the world. You can see that we have instruments implemented in every countries around the world, the US, France, everything in Europe and in Asia. And it can be used for any type of experiment all around biocontamination control in the air. To start with the product description of the Coriolis Micro, you can see that it's quite lightweight instrument, not so big. You can use it with between 5 to 40 Celsius degree. You have an autonomy of 1 hour on battery, and you can use it up to 6 hours with the long time monitoring option. You have the possibility to use it, I said, on battery with an NEMH battery. The most important is the airflow rate. You have an airflow rate between 100 to 300 liters per minute, and you collect any type of biological sample from bacteria to viruses, but also pollen, toxins, allergens, whatever. And you can use it indoor and outdoor. Regarding the technology, so the technology is a patented technology. It's the weight cyclone technology. Indeed, you have a cone where you pour with liquid, the collection liquid that can be adapted depending of what type of biological particles you want to collect. And when you start the instrument, a vortex is created inside the cone thanks to the liquid, and all the air and particles are aspirated and enter inside the cone, and the particles are centrifuged with the collection liquid on the cone. So at the end, when you stop the instrument, you have all the particles, so virus and bacteria, spores, fungi that are collected inside the liquid. To go a little bit further in the product description, so you will hear us speak about some different parts. You have the Coriolis body, and you can see that it's really simple to use, as you have only 3 buttons, a start button, a stop button, and 2 arrows to navigate inside the menu. You have an aspiration can, an air intake, so the air enters through the air intake, and you have the cone, which is screwed inside the air intake. The second instrument I want to introduce you briefly is the Coriolis Compact. It's also a patented dry cyclonic technology. Regarding the technical specification, you can see that it's even lighter than the Coriolis Micro. It's only 1.4 kilograms. It's really small. You have here an autonomy up to 8 hours with a lithium-ion battery. The airflow rate is fixed at 50 liters per minute, and you can then here also collect any biological sample, and you can use it indoor and outdoor, and you have also a Bluetooth connection which is available. Regarding the technology, you can see that it's the same technology. You have so the cone with the air intake. When you start the instrument, the air is aspirated and it goes through the cone. All the particles are retained in the cone wall. And at the end, you just need to collect the particle by recovering and rinsing the cone, so you have all your sample with all your particles collected inside the cone. Regarding the highlights, so it's a pretty new instrument, and the thing I want to highlight here is that you can collect more particles with up to 8 hours of straight sampling. The sample is compatible with rapid analysis methods such as qPCR or PCR, which is used now with the SARS-CoV-2 pandemic. You do not need to concentrate your sample as you recover by rinsing the cone with the adapted volume, and it's easy to transport, can be put in strategic places or even on a drone if you need to send it somewhere you can't go by yourself. Regarding the product description, it's the same thing. You have only a 4-button arrow to navigate inside the menu, a play button and a stop button. You have the Coriolis body, an air intake and a cone. So really simple to set up and to use. I want to add one thing before giving the presentation to Florian for the tips and tricks. It's that it's compatible with any biological analysis culturing method for sure. So you can culture bacteria if you need it. You can also analyze your sample by immunological method or genetic methods. I mentioned it with the PCR for the SARS-CoV-2. And also you can analyze your sample with a spectrometry method. And of course, in the topic of the pandemic of SARS-CoV-2, the Coriolis is adapted to any environmental monitoring of airborne viruses, such as food industry, veterinary markets, but also health and biomedical population, and for sure office spaces. Florian, I will give you it's your time to speak. So
yes.
Okay. Yeah.
Okay, so I'm a application scientist for Bertin, and today I will present some tips and tricks that our scientists have gathered to collect virus in the air with the Coriolis instrument. So as you know, virus detection in the air can be quite challenging. Viruses are often present at a very diluted concentration in the air. There's also the problem of degradation. For RNA viruses, you have to have a collection method that allows for the RNA to not to be degraded so that you can analyze it. So we have gathered some tips and tricks from our experiments with our instruments that I'm going to present here. So if we can go to the next slide. So when you design your workflow for virus detection with our Coriolis air sampler instrument, there's usually around 6 steps that you have to take into account. The first and one of the most important steps is the sampling strategy. You have to choose a sampling strategy that's adapted to the environment where you're going to perform your sampling. So adapted to parameters such as sampling group size, airflow patterns. You have to take all this into account when you design the sampling strategy. Sample collection is also important. So as I said, sampling cycle duration, airflow rate, all of this is going to have an impact on the results. For the storage, you have to transport samples in appropriate conditions to avoid RNA degradation when working with RNA virus. I'll come back on each of these steps with more details. And then there's also sample processing and sample analysis step. And finally, decontamination procedures. So for the sampling strategy, for the airflow rates, for viruses, what we recommend is an airflow rate of 200 liters per minute. This is to have an optimal collection efficiency and at the same time preserve the virus. We also have a lot of users that have used 300 liters per minute and had good results. The 200 liters per minute is really a precaution to be sure that you are not damaging any virus. So that's with the Coriolis Micro. With the Coriolis Compact, the airflow rate is 50 liters per minute. For the sampling duration, we recommend sampling at least 20-30 minute time to start having reliable results. For the device position, the main rule is that you always should start the furthest distance away from the patient and then gradually move closer. Another important rule is that the device should always be placed on the trajectory of the airflow in the room. The sampling liquid that we recommend is PBS, so phosphate buffer saline buffer. So that's the optimal collection liquid for virus-laden aerosol samplings. You can also use a culture medium such as DMEM or MEM if it's something that you have at hand. What we do not recommend is using RNA shields because they have usually a very high evaporation speed. So they are not really adapted to collection with our Coriolis air samplers. The recommended starting sampling volume is in a range between 5 and 15 ml. So for sample storage, this is a very important thing to take into account, especially if you're dealing with RNA viruses like SARS-CoV-2. So the first thing, samples should not be transported in the collection cones. They should always be transferred into appropriate storage tubes with a closed cap. They can be stored for up to 24 hours at 4 degrees Celsius. And for long-term storage, they can be frozen in cryo tubes at either -20 or -80 degrees Celsius. For the sample processing, for Coriolis micro, it's usually important to reconcentrate the liquid. So, as you know, viruses are often present at a very diluted ratio in the air. So, when you want to do analysis from the Coriolis micro output, we usually recommend a reconcentration step that can be done with a tangential flow filtration device, such as the Amicon 100 kilodalton Amicon Ultra 15 from Millipore. What you can also do is start the collection with the minimum volume of collection liquid recommended, like 5 ml, and then take a small aliquot of this, so around 150 microliters, where you can perform your DNA or RNA extraction on. For Coriolis compact, there is no need for reconcentration because it's a dry air collection, so you can resuspend in your desired volume, either by using a swab or by vortexing the cone. So for sample analysis, because the output of our Coriolis air samplers is liquid, they are compatible with most sample analysis methods, qPCR, microarrays, and various viability assays can also be performed. After each experiment, the Coriolis air samplers should be decontaminated. So for the cane, air intake, and sampling cone, they can be autoclaved, except the cone for Coriolis compact. They can also be soaked in commercial bleach solution, or they can be wiped with an ethanol 70% solution. For the entire instrument, we recommend using a H2O2 device to produce a vapor to decontaminate the instrument. Ozone treatment can also be used, or alternatively, they can also be run with an ethanol-filled cone for 15 minutes at maximum speed. This will generate an aerosol that will decontaminate the instrument. So now I'm going to briefly let Sophie present the agenda for case studies.
Yeah, so for the case studies, we will have 4 case studies that will be developed by Florian. The first one is the SARS-CoV-2 detection with the Coriolis micro air sampler. We have also results coming from other teams around norovirus detection in hospital. We have one case study around the swine influenza in pig farms. And the last one is the result we have with the Coriolis compact, which is the evaluation of decontamination procedure in a dairy factory. I want, before letting Florian go through all the topics around the case study. I want to highly thank the author and the team that published the SARS-CoV-2 results. They allow us to share the results with you, and they give us access to the preprint, so we can present you everything today. I would like really to thank them for this sharing, and we know that during those times it's really important to share everything we know about SARS-CoV-2. So Florian, I will give you back.
Okay, thank you. So the first results were very exciting about presenting to you are SARS-CoV-2 detection results that were performed in London during the peak of the COVID-19 pandemic by Imperial College London. So this study was performed with Coriolis microair sampler, and the objective was really to evaluate SARS-CoV-2 surface and air contamination. The sampling design was pretty straightforward. Surface and air samples were collected in 8 different sites, including several clinical areas and 1 public area at a northwestern London hospital. In each area, 4 air samples were collected, and surface samples were collected by swabbing 25 square centimeters of items. For the collection protocol, so the sampling was carried out with the Coriolis microair sampler at an airflow rate of 100 liters per minute for 10 minutes. So that corresponds to collecting 1 cubic meter of air. And the collection liquid was 5 ml DMEM, so a cell culture medium. The analysis method were RT-qPCR and viral culture. So this is just to show you. So this figure represents the daily number of COVID-19 patients in London, and the gray box corresponds to the period of time where the study was carried out. So as you can see, it was really during the peak of the COVID-19 pandemic in London. So now the results. So the researchers from Imperial College were able to successfully detect SARS-CoV-2 RNA in air samples. So you can see here the PCR results. When for each area, when both the air sample and the surface sample, the PCR results detected SARS-CoV-2, it was declared positive. And when one of the two was positive, then it was declared as suspected. So viral RNA was detected in 50% of surface samples. It was detected in 38.7% of the air samples. The PCR results showed a viral RNA concentration that was too low for the virus to be cultivable, and this was later confirmed by viral viability assays. One important conclusion of this study is that they were able to detect viral RNA in public areas of the hospital. So the SARS-CoV-2 RNA is not only present in the patient room, but also in public areas. So the finding of these studies show that you can successfully detect SARS-CoV-2 with the Coriolis microair sampler. And this type of results can be used for hygiene practice, such as determining the use of PPE in hospitals and the safety rules when handling this virus. So now we're going to present the next study. So this one is on a different virus. This one, the researchers were interested in noroviruses. So as you may know, noroviruses are a big risk for patients in hospitals. They're responsible for 50% of gastroenteritis outbreaks worldwide. So the goal here was really to investigate the presence of norovirus G2 bioaerosols during gastroenteritis outbreaks in healthcare facilities. So the sampling strategy here was also quite straightforward. So samples were collected at 3 different types of places, 1 meter from each patient, in front of the patient's room, at the nurse's station, and 48 samples were collected in 8 hospitals during outbreaks. The collection parameters were 200 liters per minute during 10 minutes in 15 ml of PBS. For the sample processing, the sample was filtrated and concentrated with an American device from Millipore. And the analysis method was viral RNA extraction, followed by RT-qPCR. So these are the results of this study where they collected norovirus. So as you can see, they had almost, more than half of the samples that were collected in patients' rooms were positive for norovirus G2. The result can be seen in genomes per cube meter. They were also able to detect it in nurses' station and in always common areas. So this shows that during an outbreak, the norovirus is also present in public areas in the hospital. The number of genome copies detected go from around 10 to 2,000 genome copies per cube meters. So this shows that you can use, again, so the Coriolis air samplers to evaluate virus contamination in healthcare facilities. So now we can move on to the next study. This one is in a different setting. So this is a veterinary environment. The virus of interest was swine influenza A virus, which is also something that is currently in the news. So very interesting study. The motivation was to understand the transmission of swine influenza through aerosols. More specifically, researchers wanted to determine the relationship between airborne swine influenza detection and the number of infected pigs inside pig farms. So for this study, the swine influenza detection in air samples was performed in rooms that were hosting pigs of 2 different kinds. So some types of pigs had what we call maternally derived antibodies, MDA, and other pigs did not have these antibodies. So now I can tell you a little bit more about the sampling strategy for this study. So 33 piglets without maternally derived antibodies were assigned to 3 independent rooms. 33 piglets with the antibodies were also assigned to 3 independent rooms. And so the idea was that in each room, you had 2 pens. In the first pen, you had 2 cedar pigs that were inoculated with swine influenza H1N1. You had 4 pigs in the same pen, so that were in direct contact with the cedar pig here in red. And on the other pen, you had 5 pigs that were in indirect contact with the cedar pig, so in a different pen that was 30 centimeters apart. So for the sampling strategy, so the Coriolis micro air sampler device was placed between 2 pens, 7 centimeters away from the ground, so at the height of the pigs, but without direct contact with them. Nasal swabs were also collected. The collection parameters for the Coriolis device was 300 liters per minute during 10 minutes in 15 ml PBS with 0.005% triton, 3 times a week for 25 days post-infection. For sample processing, so once again, the sample was filtrated and concentrated using an American device, and samples were analyzed with viral RNA extraction followed by RT-qPCR. So now we can look at the results of this study. So on the left side, you can see the SWINE influenza genome detected in air samples with RT-PCR analysis. So SWINE influenza genome was detected in aerosols from all rooms, starting at 2 days post-infection and with a peak at 9 days post-infection. On the right side, you can see the results of the nasal swabs. So all indirect contact peaks were shown to be infected. So this shows that you indeed have aerosol transmission of the swine influenza virus, which is confirmed by the air sample results, which show you the swine influenza genome load in air samples. So this study shows that you can also use the Coriolis micro air sampler in a veterinary environment, and this can give you more insights in trying to understand the virus transmissions through bioaerosols. So the next study that we're going to present is in again a different environment. So this time in the food industry. So as you may know, in cheese production, there is often issues due to phages infection. So phages are species of virus that can infect bacteria. In this case, so this was performed in a dairy factory, and the problem was phage infection of lactococcal bacteria. So that can hinder fermentation processes and cause issues in cheese production, such as variation in taste and texture. So the goal here was to evaluate the impact of decontamination method using ozone on the bacteriophages present in the air in a dairy factory. So for the protocol, so first the decontamination was performed using an Alphatec mobile disinfection unit. The ozone concentration target was 2 ppm per 2 hours during 8 hours. For the sampling strategy, so for this study, the Coriolis compact was used for 5 hours at 50 liters per minute. For different sampling, sampling was carried out at 4 different times with also a control sample. So first before ozone treatment, then just after the ozone treatment. The third one 2 days after the ozone treatment, and the last one after a second ozone treatment. For the analysis, so DNA extraction was performed on the Coriolis compact samples, and then simplex PCR specific for the Lactococcus lactis phages, with a limit of detection of around 100 phages per milliliter. So now we can show you the results. So these are the results for different species of phages, C2, ASK1, and PP35. So as we can see here, there was an important impact of the ozone treatment on C2 bacteriophages. So each time when we collect sample after the ozone treatment, we can see the concentration of C2 phages is really much lower. On the other hand, the ASK1 bacteriophages population seems much more resistant to the ozone treatment. The second ozone treatment has almost no impact. So this can be due either to a higher degree of contamination by ASK1 phages or just a higher resistance of these species of phages to ozone treatment. So this study is really to show you that you can use the Coriolis compact successfully to detect virus. It's a very good instrument in the context of the food industry because it's very easy to carry and to use with a battery. And these type of studies can be used to evaluate the impact of decontamination procedures, which is something that will also be relevant in the future, especially in the context of the COVID-19 pandemic. So just to conclude, I'm going to let Sophie present the conclusion.
Yeah, so thank you, Florian, for all these case studies. So before the Q&A session, I just want to give you back all the highlight of each instrument. So as you understand, it's really innovative and comprehensive system, easy to use. The first one, the Coriolis micro air sampler, the biggest advantage is a high airflow rate. It's also be combined with a long time monitoring platform to perform longer sampling, and you have your sample in a liquid output compatible with molecular analysis. For the Coriolis compact air sampler, it's easy to use and to transport on the field. You see with the case study in the dairy factory that it's really easy to put in place, and it's a dry sampling patented technology with no resuspension step before molecular analysis, so you have access to a concentrated sample. And those two instruments are idle instruments for airborne viruses detection. And so you have here all my contact, and I would like to thank you for your attention, and we will now go through the question and try to answer a maximum of questions. So first thing, I will answer to several questions we had in the question box regarding a more technical point of view. So you will have access to the presentation and the recording of the session. So at the end, the session was recorded, and so at the end of the session, and I think in the week, you will receive an email with a link to download the recording and the presentation. So you will have access to all the data that we present this afternoon with Florian. So Florian, if you are ready for the first question, I have one, so which is pretty interesting. It's what influences the choice between Coriolis Micro and Coriolis Compact, and perhaps if you can add a few words about specifically the virus detection.
Yes. So I think choosing between the Coriolis Micro and the Coriolis Compact, there are several parameters that you want to take into account. The Coriolis Micro has a higher airflow rate, so you can have a smaller air sampling time, which can be useful in some situations, for example, in healthcare facilities where you might not want to leave an instrument in a patient room for too long. On the other hand, the Coriolis Compact has higher battery autonomy, which can be really useful for on-the-field collection. And also, with the Coriolis Compact, you are doing dry collection, which means that you can resuspend the sample in any volume of liquid that you want. So you avoid all the reconcentration step and potential losses of material, which can be really useful when you want to analyze your sample, for example, with RT-PCR. So I hope that answers your question.
Thank you, Florian. So next question, we have something, we have several questions around the decontamination. So for decontamination, is it 100% ethanol or 70% ethanol? And how do you have H2O2 vapor? So it's several questions. I try to have it in one question.
Yeah, sure. So it's 70% ethanol for the decontamination. And for the H2O2, so what we recommend is using a device that can generate an H2O2 vapor, which can be purchased. So it's the same type of device you can use for incubators. An alternative is also using ozone. So you can use ozone to decontaminate the Coriolis air samples.
Okay, thank you, Florian. So The next. So the next question, really interesting. So the majority of viruses are less than 0.5 microns. Therefore, how efficient is the Coriolis micro to collect viral particles? And what is your feedback on that?
So that is a very interesting question. So as you may know, viral particles are very rarely naked in the air. They are almost always present with other particles in a bigger particle. And usually the diameter of this we call virus-laden particles is higher than just the virus by itself. So there are a lot of particles that are in the micron range that can be collected easily with the Coriolis air samplers. To give you an estimation of efficiency, so for particles of 0.5 micrometer diameter, we have a collection efficiency of 50% at an airflow rate of 300 liters per minute with the Coriolis micro.
Yes. So if I can add, there is paper available that show that the majority of the virus, as Florian said, are not naked and alone in the air and are always collapsed to dust or anything else. And so it's above 0.5 micrometers.
Yes.
The next question. So you begin to answer, Florian. What is the physical collection efficiency of this device? So you said 50% at 0.5 microns. And so do you have the numbers for bigger particles?
I do not have the numbers with me for bigger particles, but it is something that we can look to send you later.
So it's around 90 to 100% for particles more than 3 micrometers and up to 10 micrometers. We have a question which is linked to that is, do you have a maximum size of particles when you collect with the Coriolis micro? So I can answer that directly. We do tests with physical particles up to 20 micrometers, and as for biological purpose, we estimate that it was the maximum size of the particles which can be analyzed. I have a question regarding, I have several questions regarding collection liquid. So do you know if there is specific virus that require Triton or not? And do you have any other information? Because I had a question regarding the PBS. Is the PBS recommended for long monitoring to avoid?
Okay, so I will answer the first question. So usually for virus, we do not recommend adding surfactants such as Triton to the collection liquid because they might interfere with the virus envelope. So we recommend just using PBS. And the second question, for long-term monitoring, the recommended media is also PBS. Even though you can also use cell culture medium, PBS is really the chosen medium.
Okay. I have another question regarding quantitative and qualitative results. So what is your feedback on that as you are speaking about SARS-CoV-2 and qPCR?
Yes, so you can obtain quantitative results, as you could see, so in terms of genome copies per Q meters. That being said, for the Coriolis micro, you have this reconcentration step. So it's possible that the results that you obtain is actually underestimated due to loss concerning that. So with the Coriolis compact, you can avoid this, and we have data to show that you can collect quite a high quantity of the virus with it. So I don't know if that answers your question, Sophie. Do you want to add something to that?
No, it's okay. Let me just go through the question because we already answered to some of those. We have question about similar product in the market. So, of course, I will start the answer, and Florian, if you want to complete at the end. So, I can say that there is other air sampler on the market, but we mentioned at the beginning of the presentation that for Coriolis Micro and Coriolis Compact, it's a patented technology. So Bertin technology is the only one to provide this type of technology and cyclonic technology. And so, of course, you can find other air samplers, but not with such efficiency for virus, specifically for virus collection.
Yeah, so if I could add something, so a large part of the other air sampler device on the market, they use filters. So as you may know, filters require a lot of manipulation steps to be used. So that can be complicated and even a little bit dangerous in the case of virus. The advantage of the Coriolis air sampler is really that you avoid all these manipulation steps because your viral particles are already in the liquid. You do not need to manipulate anything else. So that can be an advantage.
Yeah. So we have a question. If I go back to the results, do we have case studies or feedback from users which recovered light virus using plaque assay from Coriolis Micro? Are all the studies detect viral genetic material?
No, we do have some studies that that use viability assays. I can send you some example of studies we have in the literature if you want. No, it is something that has been done. Yes.
Oh, we have a question regarding ethanol is to kill bacteria or virus, how to remove contaminated DNA or RNA in the instrument, any results to show the effectiveness. So I think we already go a little bit through that.
Sorry, I did not hear the question. There was a connection issue.
Sorry. So ethanol is to kill bacteria or virus. So how to remove contaminated DNA or RNA in the instrument? So you mentioned already a little bit of answers here.
I think for the instrument, it can be decontaminated with ozone or H2O2. That's probably the better way. For the cones and the cane, the air intake, you can use autoclave.
Another question about media, collection media. Are there any example or cases of using Coriolis Micro collection liquid directly for PCR? Any effect of PCR inhibitor issue?
Yes. So in the case of the SARS-CoV-2 study that we have showed you, they have performed PCR directly on the sample from the Coriolis. So in the case of DNA extraction, you have steps where you are basically washing the pellets, so you can take away all the PCR inhibitors, and it should be okay to use. For the RNA extraction, we have results that show that it's also working well. It is preferable, though, like to use the filtration and reconcentration step if you can.
So we have a question which is two questions linked together. We begin to answer, but apparently the answer was not complete. It's regarding the collection, the long collection with the Coriolis micro. So one question, can we collect more than 6 hours with the Coriolis micro? And is there any effect about PBS as it will provoke an enrichment of salts in the samples? So the question is, yes, you can collect more than 6 hours with the Coriolis micro, but you will need to launch 2 cycles in a row, and you have right, in case of very long sampling, a question of liquid is asked, and we need to discuss about how long do you want to sample and how fragile is your sample in order to avoid too much salt. And in this case, perhaps water will be preferable and will be a better balance in order to have a better efficiency. So Florian, do you want to add something for this question?
I think you're pretty accurate. For long-term sampling, you can also use the Coriolis compact, and in that case, you avoid all problem of wondering about the salt concentration.
Yeah, and I think we still have few minutes, so I have another question regarding Coriolis micro. Can viruses be removed in the evaporated liquid? So do you have feedback on this? How the cross-contamination can be?
You said can
regarding
something be removed from the liquid?
Yeah. Can viruses be removed in the evaporated liquid?
You mean, can you have loss of material due to evaporation? So the answer is
yes.
Yes, you can. Which is why it's better to start with the large enough volume of liquid so that you do not have too much evaporation.
Okay. We have no more technical question right now. So I want to add again that you will receive an email with the link of the recording, and also an email with the link to download the presentation. Another question was, if you need, we can send you e-certificate as you participate at the presentation. So if you need it, just send us an email and we will send you back the certificate. I would like then, before shutting down the session, to thank you for your attention and your interest. And of course, Florian and I and all Bertin team stay available for you. And don't hesitate to contact us after the session if you have any other question which pop up. So thank you all, and depending where you are around the world, have a good day or a good afternoon, and see you soon, I hope.